Okay, good evening, everyone. And we will start about a new lesson or new part of the syllabus that is about analysis of gene function. We have learned about analysis of genetic variation. We have also learned about analysis of gene expression. There might be here and there a little bit overlap or mix up, but still you should be able to know now about how to quantify the amount of mRNA or RNA expressed, how to quantify the amount of protein. Probably that we will also have a look at it as a part of this one. But otherwise, Western blotting, Southern blotting, you can use those techniques, RT-PCR, and so many other techniques we have learned. We also learned about for genetic variation. We learned about Southern blot. We learned about microarray technology, gene libraries, which could be also will come between cloning and these things, screenings, and so on. So now we are going to learn something more interesting, that is about gene function. If somebody gave us a piece of DNA and asked us, tell us what is the significance of sequence information, tell us what is the outcome or phenotype of the information. You can say it as genotype, right? That is the gene, and what is the result of it. Whether it doesn't matter if it is working as an RNA molecule or if it is working as a protein. That is how we should be able to find out. There are so many genes in the genomes of many species that we don't know what their function is yet. And imagine in the ancient times, when they were not ancient means in the earlier days of when they were trying to find out a gene function, how did they come across? How did they solve these things? So there are two words that we should try to understand. This is a good one. One is called as forward genetics, and the other one is called as reverse genetics. Usually, if there is a person, say about the person's height or tallness, we observed a phenotype. That is a phenotype. And we need to find out, or first observed things are, of course, phenotype. We have been doing that for thousands of hundreds of thousands of years, since the evolution of brain to think about many other things. But we didn't know what is the molecular basis of it. We never went to that. Now that we know about DNA, we know about genetic material, we know that majority, if not all, the consequence of every phenotype is coming from the gene, right? So an individual or a cell, everything is between two things. One is the genes that the person has, and the other one is the environment. Or you can say, I hope you heard the nature and kind of nurture. Nature is the genetic nature of the person. Nurture is how it is influenced by the surroundings, right? We are accumulative. The way we behave, even, or the way we think, also has molecular basis as well as environmental factor. Both are responsible for the way we develop. And even in a cell, how it responds to environmental things is again based on the genes that are there and what kind of stimulus is being received from them. Okay, so what I'm trying to imply here is we usually, traditionally, we were able to see a phenotype, and then now we are asking what gene is causing that phenotype and how is it causing it. So if we are going from a phenotype, this is a phenotype, and we are trying to find the gene responsible for that phenotype, then we call that as forward genetics. There is also another issue is with bioinformatics and with lot of sequencing. Okay, and we have to also discuss about sequencing, that we will come in a while. In a few classes we will discuss about it. With bioinformatics and sequencing, we know more genes, but we don't know their function. We don't know what is their phenotype. There are, just take E. coli, there are about 800 genes for which we don't know, or open reading frames at least, for which we don't know if there is a function or not. So if we are going from nucleotide sequence and trying to find a function or the phenotype of it, we call that as reverse genetics. Okay, so these are two approaches you want to remember. If I told that a fungi is giving, a fungus is giving, has amylase, that is a function. It is the ability to digest starch, say. And I want you to clone the gene and then characterize the gene and tell me the nucleotide sequence. You are going from function, from the phenotype to the gene. So it is a forward genetics. Reverse genetics is little tougher. Somebody has given you a gene's nucleotide sequence and asked you to find out how it is done. So these two, if you want to find a function, usually then it is actually a difficult way of doing things. There are several strategies, and just as an explanation, or the strategy wise, if you see, there are called as genetic screens, something like the screening we have learned, which we will discuss about it in a few aspects. In the sense, we have a, say for example, we have a phenotype, and we will try to get an organism or a mutant in which the phenotype is missing. Right? That means something must have happened to that gene, isn't it? And then we try to identify the gene that has been mutated. Right? And then we can know from the phenotype, we are able to determine the gene, which gene is responsible or has a role to play in that particular phenotype. In reverse genetic screens, the strategy is a little bit different. You, as I said, the sequence is given to you. So what we do is we delete that gene, or we know the gene, so we delete the gene. Deletion, and then see what is happening to the organism, right? So there could be a change in the phenotype, there could be lethality, there could be something. We will try to identify what is the phenotypic changes because we have deleted a gene or mutated a gene. So that is how these forward genetic screens and reverse genetic screens are become useful to us. And it becomes, in my own experience too, there are several genes in which we deleted, there are some deletions, but we can't see any noticeable phenotype, right? That makes it a little difficult to find out. Not everything is visibly it's showing up. Okay, so that makes it a little bit difficult. So there is one more thing I want you to also think in these lines. Every gene that we know of will produce either RNA, from which RNA may produce protein, right? And maybe RNA gives a function or a phenotype. Maybe at the level of protein as enzymes or as structural proteins, they might give rise to a function or a phenotype, right? These are two, both are possible. If they are giving a function, one thing that we have to understand is either of them, they must be interacting with something else, right? They will interact with other, it could be RNA, it could be other proteins, it could be interaction with the DNA. They are interacting with some molecule. It can be a ligand, that is a molecule like within the cell, or it can be an enzyme. Enzymes also bind to substrate, right? Everything is about interactions. All life is similar to what our society is, human society. Individuals, you can take a person as an individual. And how does an individual perform? Say an individual, a teacher will speak to several others and do some things to them, right? Change, transfer of knowledge, and so on, right? There is interaction. The teacher outside in the classroom may be interacting with colleagues. That is an interaction. The same way, a protein, assume this is a protein, will interact with some other components and may transform them into something useful. And every function, whichever type of function it is, there is going to be interaction, right? Interacting with RNA, protein, DNA, or ligands, or as an enzyme. That is what we should try to find out. So for that, we will see if a protein is interacting with DNA, how can we identify it? So the question here is, it's called as gel shift assay. Okay. Assay. You can also call it as enzyme mobility shift. That is much more correct. Mobility shift assay. It's called as EMSA. Right? Enzyme mobility shift, change in the mobility. Where is it? It is the electrophoretic mobility assay. So here is the technique. We take the DNA that we suspect that it is interacting with, it is probably that is where the protein interacts, and we take that DNA and we have to radio label it. Radio labeling is done by end labeling. How do you do end labeling? It is using polynucleotide kinase that will put 5' phosphate, right? The phosphate could be exchanged. You have to provide ATP. The gamma phosphate of ATP will be transferred here. So what we should do is we should have used gamma P32 radio labeled ATP as substrate. You add the polynucleotide kinase would use that as substrate and transfer the P32 here, radio labeled one. Okay. So that is how end labeling can be done. So if we had DNA and the DNA is, and we are going to make 4 different tubes. And in one tube, we are going to have only the DNA, the gene that radio labeled DNA like this. In another one, we are going to add DNA plus little bit of amount, say 10 micrograms. I'm just making it up. In another one, we are going to use DNA plus 20 micrograms of DNA, of protein. And in the third one, we are going to use DNA plus some 50 micrograms of protein. We'll allow the interactions to happen, and then we will load them in the sample at that same time. And then we will connect the electrodes and then allow the electrophoresis to happen. The one that is free of any protein will run faster. In this case, we are finding 2 bands because we are actually using, say, for example, if you allow me, I would try to rephrase it here. So I'll talk in terms of molecules instead of proteins. I have taken 100 molecules of the gene or DNA, okay? and 0 amount of protein. In the second case, in all the cases, I'm using 100 molecules of the DNA. In the protein, I'm increasing. In the first case, I have given, say, 10 molecules, or say 20 molecules. In the second one, I have given 40 molecules. In the third one, I have given, say, 120 molecules. Then what would happen? In the first one, there is no protein, there is no molecular weight increase, so the DNA will run at a certain level. Whereas in the second case, where we have given 20 molecules of protein and 100 of DNA, so 20% of it will run slow because the DNA is bound along with the protein. The rest of the 80 molecules will just travel as per the regular molecular weight. In the other case, we have added 40. So you can assume that there are 40 molecules here and some 60 molecules of DNA here, right? And here we are adding 120 molecules of protein. So all the DNA molecules are likely to be bound by protein. So all the 100 DNA molecules are here. You see that there is mobility shift. The mobility should have been here till here, but then there is a shift. So we call it as enzyme mobility shift assay. It's also called as gel retardation assay. Retardation is about mobility, retardation in the mobility. Some people also call it as gel shift assay. Okay. So I hope you understood that. If there is no interaction, say for example, I'll try to cook up a different scenario so that probably you can understand better. I have given you one DNA molecule, right? And I'm giving you two different proteins, protein A, and I'm giving you protein B. I can ask you, tell me if, or you can think of it like protein A is binding to the DNA or not. Maybe let me try. So I'm giving a situation where you're going to have to figure out which of these two proteins bind to the DNA or not, or are they binding at all? That is how you can also think of it. So how would you set up an experiment? You will have several of these kinds of things, and then you will find out one second. If you give, I'll just figure it out. Right? Yes. So now I will make several tubes, and in each of them, I will put DNA molecules. The DNA is this. I will say in all of them, I have 100. And I'm going to, maybe that's enough. And in this case, I'm not adding any protein. So 0 amount of protein. In the second case, I'm going to add 20 molecules of A, 50 molecules of A, and 100 molecules of A. In the second set of experiments, I will going to add 20 B, 50 B, and 100 B. Now, if I had the result, the bands are looking something like this. This is where the naked DNA without, in which we didn't add any other proteins, is running so far. And then we will find something like this. What does that indicate? Yes. What does this indicate? Anybody? What does this fragment say? This is just the empty DNA, only DNA alone here, and it is running at this pace. And as you see, with the increasing amount of protein, you see that there is a reduction or in the enzyme mobility shift. So what does that say? Protein A is interacting with the given DNA sample, right? That is the interaction. In protein B, we do not find anywhere that there is interaction because there is no shift. If there was interaction, one would have expected that there would have been some of the bands here as well. Okay, that is how one can say whether the given protein sample is interacting with the DNA or not. I'll also give you a little more a little more twist in this. Say my goal is to identify where exactly in the DNA. Say, for example, this is the length of the DNA, and I want to know where exactly the protein binds. Does the protein bind here? Does the protein bind here? Or does the protein bind here? How can you figure that out? So I'm going to, for convenience sake, separate this DNA into 3 different parts, okay? Just for representation purpose, X, Y, Z. Say in the previous experiment, we found that protein A is interacting with the DNA. So protein A is something like this, sorry, DNA is something like this long, and protein A is interacting. We don't know whether it is interacting here, here, or here. And that's the goal we are trying to use, trying to find out how gel shift assay can be used to do that. So in this case, what we will do is we can design new DNA molecules. DNA molecules which will have this much fragment. So this is a DNA fragment, and I'm taking another DNA also something like this. One of it has X and Y, right? X and Y regions. The other one has Y and Z regions. This is just for, it is a nice way of doing things as well. So if we can construct a DNA. Can anybody help me trying to do this? And the objective is to find out whether protein A interacts in the position X, position Y, or position Z. You can do it simply also, but sometimes if I just gave only X and only Y and only Z, it might be too small for the protein to interact. So in that case, interaction may not be possible. So here what we are trying to do is, without compromising that, how can we figure out? It's a fun way of deducing where the protein might be binding. And that's why I intend to take this up. Okay. So we need to have several control. Always, every time we are doing, you should use a control. Anything with a control is an experiment. Anything without a control is not an experiment. In this case, we are the first one that we are used, where only DNA and nothing is given. That is a control. We can also use a control like I have DNA molecules are given. I know a non-specific binding protein, DNA binding protein. It's something like histones, say, for example. I'm just making it up. Histones will bind to DNA. They will recognize the phosphate backbone, deoxyribose, and they will interact. So if I added histones, then the DNA, say, for example, I added 100 histone proteins. It's just a theoretical one. In that case, it should show like this, right? Then I can say that A is interacting and B is not interacting. Okay. Anyway, in this case, I'm going to use X and Y together as DNA fragment, Y and Z as DNA fragment as well. So I have only one protein. Protein is A. I might have, say, for example, I will just simplify it, okay? In this case, I'm going to add X, Y, Z together, the whole fragment. I'm going to have X and Y only as a fragment. I'm going to also have Y and Z as a fragment. no protein added, right? We can also have, I will just put it Z, XYZ complete, and then I'm going to add XY, XY, YZ, YZ. I hope you are following it enough.
Yes, sir.
Yes. So I'm going to add protein now. In this case, I'm adding 100 protein A. I'm not adding any protein here. Okay. I'm not adding any protein here. I'm going to add, I'm not adding any protein in XY. I'm going to add protein A. I'm not going to add any protein there. And I'm going to add protein here. These are the lanes. Okay. Those are the samples loaded. So XYZ has a certain molecular weight, so it will run as fast. If we already knew that the protein A is interacting with the DNA in the previous experiment, theoretical one that we have done, so we will show that the band there should be a shift, or gel retardation should have occurred, because if A is interacting with XYZ, the whole DNA here, we want to figure out whether it is binding to X part, Y part, or the Z parts, theoretically. So we constructed two DNA molecules, XY and YZ, and then we are trying to check. So here in lane 3, we have only XY. So it is smaller, so it should run a little faster like this. Now in the lane 4, we have A plus XY. If there is interaction, you should find it somewhere here, approximately. And the next one is lane 5, in which we only have Y and Z. Assume these two XY and YZ are same molecular weights in length or so. So you'll put it like this. And now we have YZ. If I have YZ and A is interacting also, then the shift will be approximately here. If I showed you a graph like this, a gel picture like this, what can you deduce out of it?
That gene Y is interacting with protein
Yes. So the option is we are not calling gene, we'll just say part or fragment of the DNA. Whether A is interacting with X, or A is interacting with Y, or A is interacting with Z. That's the question. If you find interactions in this case, right, in lane 4, this is your control, because we already know that A is interacting with XYZ DNA, right? This is a control. So with because using the control, and now we have seen the mobility shift. So A is interacting with XY. And from lane 6, you also can see that A is interacting with Y and Z. And the region common between, that means it is interacting with both these fragments. That means this is the common part of it. Okay. So you can assume in that case that we are the DNA, the Y region of the DNA is being interacted by the A, or A is binding to Y region, right? That is a nice way of how experimental design is done. One thing I want you to keep in mind is there are DNA binding proteins. Whether they are sequence specific or not is a question. Specific and non-specific. If they are specific, if they're non-specific, most of the interactions are with the phosphate, deoxyribose, and not much with the bases, right? They won't make any specific interactions with the nitrogenous bases. Whereas if the DNA protein that is specifically binding, They might be interacting with phosphate group, deoxyribose, and also the nitrogenous bases. What we are discussing is mostly about sequence-specific DNA binding proteins. This is used for sequence-specific DNA binding proteins. It's not as useful for non-specific bindings, but it can say, like in the previous one, you can give any different types of DNA and then say if whether it is sequence specific or non-sequence specific. If I said A is binding to XYZ DNA, it is unlikely to bind to a region which is a PQR, which means that sequence is entirely different compared to this one, right? Then A protein is sequence specific. Like in the other case where if we added histones, it will bind to any kind of DNA that you give. There is no problem at all. So that is one more thing you need to keep in mind. Okay, why go through these many things? We can also have another method that is called as DNase 1 footprinting. It is to find out where exactly can you have DNA binding protein. DNA binds to the protein, protein binds to the DNA. In this case too, we will have end labeling, and we will figure out how to do that in the later class maybe. In here also I said we will do end labeling, right? We will do end labeling. So we have two ends, 5'. We are using double-stranded DNA here. This is also 5', and this is 3', and this is 3'. If we add polynucleotide kinase and provide the substrate of gamma p32 ATP and allow the interactions on both the sides, we will get radio labeling, right? Both sides, the labeling is done. And there is no issue. It will not compromise our experiment. The next one that we are doing actually is DNase 1 footprinting, which would require that we only have to have label on one of the sides. We only want, we only want radio label only on one side. How can we get a DNA that is labeled only on one side, N labeled on one side? Anybody? You can think of any possibilities, and whatever you're thinking is correct. Don't worry about whether it'll be right or wrong. We can come to the answer eventually. I only want labeling on this side of the DNA. I don't want any labeling on this side. How can we do that? If I take it as such and add P and K and give all the ATP, everything, it is likely to label on this side also. I want to prevent it. How can we do that? Yes. There are many ways. If you had at least I want to discuss for the sake of understanding, okay, and thinking, so that we can probably think on those lines when it is required. If we had a situation of DNA like this, and there is a restriction endonuclease at the end, okay, like this. And in that case, I could add polynucleotide kinase and ATP, and both these sites would be labeled. And then I could have used the restriction endonuclease and gotten rid of this part. That is one way of doing it. That is also, it's complicated, but one way of doing. The best way to do is something even better on. So what is done in that situation is you take a template DNA like this, and then you would synthesize, you would have used primers, and the primers, so this is 5', and this will be 3, this is 5', and here is 3. So I should have a primer in here. So it will be, one second, I'll just adjust it. This is 3'. So the primer will have 5' and 3' OH. The other primer will be here, which will be 5' and 3', right? I hope you agree with that. Now, before the start of PCR, I would label this primer, the one that I'm going to mark. I can label this primer. It is single strand DNA, right? Oligonucleotide. It has 5' and a 3'. I could treat it with PNK, polynucleotide kinase, provide the ATP with gamma labeled P32, and what will happen because of it is that the primer will get labeled, and use the primer here. Now this is labeled one. Use that primer and make DNA. If you allow the PCR to happen, right? What will happen? All the products that are formed will have DNA, something like this. The products that will be like this, and say this is 3' and this is 5. You'll have the radio label here. That is how we can do labeling only on one side, right? Did you understand this? Any questions here? So these are important points. Yes, please go ahead. You said it's clear, or
Yes,
you had a
I said it's
question?
clear.
Okay, that's fine then. Thanks. So,
Sir,
yes, please.
sir,
Go
the
ahead.
second primer will also be radio label, no, sir? Because that is also 5 dash 5 prime to 3
Yes,
prime.
but we get primer 1 forward and reverse in separate tubes. So we are taking only one of the primers and doing the end labeling. We are not doing this anything. After labeling it, then we are going to add it to the PCR mix with the forward primer, reverse primer that is labeled, template, DNTP polymerase, and then perform the PCR. Okay, there is nowhere this primer is going to come into the reaction mixture at the stage of end labeling. Is that understood?
Yes, sir. Thank you, sir.
Okay, chill. Okay, now what we are talking is why did we make this end labeling, and why only one side of the DNA we want is the question. The objective of this DNA is one footprinting is to exactly know where in the DNA the protein binds. This experiment that we did using gel shift assay or enzyme mobility shift assay provides information, but it does not exactly say which bases the DNA is interacting with. The protein is interacting with, right? So in that case, DNase one footprinting is very useful. So we take DNA, we have labeled it as the procedure, as per the procedure given here, and we allow the DNA to interact, we allow the protein to interact with the DNA. And then we are going to add DNase 1. DNase 1 is just randomly, it will cleave all the DNA, whichever is accessible. But if the protein is bound, if the protein is already bound to the DNA, those regions will not be accessible for DNase to act upon, because DNA is already occupied by some other protein. So that region will get protected, or whereas all the rest of the DNA is going to get digested. What you should also keep in mind is it's not that we are going to allow complete digestion. It's not complete digestion, it's kind of partial digestion. If you perform partial digestion where DNase might act on some places and may not in some places, and you are adding so many of these molecules, right, DNA molecules along with the protein. So now let's see what all will happen. This region is there, and this region will also be there. Will be there. You will have several regions cut and uncut also, maybe, right? What we are going to do is, of course, electrophoresis, and then followed by radiography. The previous one also has the same thing. Okay, that is where we are using the end labeling for autoradiography. So we perform electrophoresis. Sample 1 is control, that means no protein is added. And if no protein is added, you will get several fragments, some things like this, right? And this is the test in which we added the protein, and we see that some fragments are missing, like in this case. And I want to complicate this. Let's complicate it. If I have labeled on both sides, what would happen? If I have labeled on both sides, this fragment will also have label. This will also have label, right? The remaining cut DNA. And the gel would, upon development, the gel would look everywhere something is already there. It will become complicated. We want to keep track of it. So what we do is, and that is the reason why we are only labeling on one side of it. So whatever will show up will only be these. Upon autoradiography, only the labeled fragments will come up and the others don't come up. And that is the reason why we are labeling only on one side of the DNA. And so what you will see upon comparison between test and the, this is the test and this is the control. Control, we did not add any DNA, so you'll find a ladder-like thing almost completely on base-wise. But wherever the protein is bound, that region, you will not find any cuts because it's not accessible to the DNase one. And therefore, that region is protected and you will not see bands in that region. And that is called as the footprint. It's like where the protein is standing on the DNA. You can figure that out. Okay. Any questions on this? Okay, we will move on. And if you have a question, rewatch the video once again, and you can come back, and then we can ask. We can discuss again. What I want to also discuss today is you must have heard about Yeast 1 hybrid system and Yeast 2 hybrid system. This is a way, another, say in the previous ones, these are experiments done in vitro. If we want to prove that a protein is interacting with a DNA molecule, we show it in vitro. What is shown in vitro doesn't have to be true in vivo. That means within the cell, what is the guarantee that it works the same way? What is the guarantee that it interacts that way? So we need to prove the in vivo, as using an in vivo assay, that the protein is interacting with the DNA. And for that, we can use something called as Yeast 1 hybrid system. This is a complex one because we will try to bring in several things into one reporter assay. So let's see what is it. The bait sequence is the sequence that for which we are looking. We assume that there is DNA is interacting with. Okay, the protein. So assume that there is a region called XYZ. That's the DNA I'm thinking. And I'm testing a protein called A, and I want to know whether it interacts with XYZ or not. So what we do is we make two constructs. One of it is having a reporter gene, something like LAGZ, for example, beta-galactosidase enzyme gene. I put this XYZ here. I'm marking it as this one, XYZ. You can have multiple copies also. And that is called as the bait. Okay. Now I want to find out if protein A is interacting with this region or not. For that, we are going to make a hybrid. What is the hybrid? The hybrid is we are going to clone gene A. We're going to make translational fusions. What is a translational fusion? We're going to make gene A, right? And we are making a translational fusion with GAL4 activation domain. GAL4 is a transcriptional factor for galactose biosynthetic pathway, I think. And it has two domains like this. it has a DNA binding domain and it has activation domain. Activation domain is responsible for bringing in RNA polymerase and other things and allow the activation of the transcription from the gene here. DNA binding one is responsible for interacting with the promoter or operator regions. Okay, so what happens is in normal scenarios, this is the gene for say galactose biosynthetic pathway, and GAL4 using its DNA binding domain, it will recognize the promoter operator elements, and with this activation domain, it will bring in the RNA polymerase and allow transcription. So we are going to make a different scenario. We are going to replace this DNA binding domain. We are going to remove that. And we want to find out if it can be, we want to make something like this. Can we make protein A and C, use it as a DNA binding domain? That's the point. So what we need to do now is, I hope you understood it. Let's see. If I want to test this reporter system that is present here, I need to do something. I should somehow be able to bind to the DNA XYZ, and I should be able to activate the transcription. So I need AD, that is the activation domain of GAL4. We don't know, protein A does not have anything. All we know is it interacts with the DNA. We don't think it is a transcriptional factor, but we want this reporter to be expressed and a protein be produced. So we are going to use this A as a DNA binding domain, but A interacts with XYZ, right? So we are putting a bait sequence here like this. And if the interaction is like this, a activation domain will allow the transcription of LAGZ, and then we will get the gene for LAGZ. Okay, that is the thought process in that. I will only repeat this once again so that, and also in the next class, don't worry if you have any confusion, because you need to be very clear about it. And because there is also Yeast-2 hybrid system that is coming up in the next classes. So here we are going to make a transcriptional. Why is it called one hybrid system? We are making one hybrid here. We made protein A, and we are using GAL4 activation domain. So when it is produced, it should produce A, that is the protein of our interest that we are checking, and the GAL4 activation domain for activation of the transcription of the promoter region, the reporter gene. Okay. So if there is no if there is no reporter production, that means something has not interacted. There is no interaction here, right? If there is we find something like this GAL4AD with the DNA binding protein, something like A that we are discussing, then you will see a reporter being expressed, right? That is the logic of Yeast-1 hybrid system. Just to summarize, and let's see if I can try it a little bit differently. So I will give you a cell. In all the cells, we have, in this case, I made a transcriptional, the hybrid is protein C and activation domain. And in the other case, I put a plasmid with protein X and activation domain. And I put in the reporter system that we have, bait sequence plus reporter. We have it here, okay, LAGZ, and the bait is here. Same bait in both the cases, okay? And one of it is showing a blue color when grown on XGAL, and the one of it showing blue color Other one is showing white, or say creamish, not blue. So what does it mean? And how did you come to that conclusion? Anybody, please? Yes, anyone? If it is blue, it means laxate has been expressed. How can laxate be expressed? This is our bait sequence. Laxate can be expressed, and we put in a hybrid. The hybrid should have produced activation domain was here. Activation domain allowed the transcription of it. But activation domain is actually in, or it is a hybrid protein, or it translationally fused to protein C. If there is interaction here, only then the activation domain can be in this location. And only then you will have the transcription and the production of laxate, because of which we will have the blue color. In this case, we use the same bait sequence. We want to find out whether C or D interacts with the with this bait sequence. So we already made a protein that is X with activation domain of GAL4, but it is still white in color, which would mean that laxate is not produced. Laxate is not produced because there is no expression of the reporter. That means activation domain did not come here. That means X was not interacting with this bait sequence. Okay? Did you understand this?
Yes, sir.
Okay. Yeah, I'll stop here, but tomorrow we're going to read about also about two hybrid system. I think it's going to crash now. Let's see. Two hybrid system, East 2 hybrid system, which is used for finding protein-protein interactions. Protein DNA interactions, East 1 hybrid system is for finding protein DNA interactions. We'll also have several other techniques like northwestern blotting and many other things to find out DNA binding proteins or RNA binding proteins as well. Okay, we will not discuss every of them because there are whole huge number of techniques. I want you to learn about the design, the principle, and how to interpret it. And because the same things, little bit deviation, and you can find out, you can say what it means. Okay. Any other questions? Anything to discuss? Yes, please.
For now, no, sir.
Okay. Then good. Thank you very much. Thank you for your attention and coming for the class.
Thank you, sir.
Thank you, sir.
Yes. See you. Yes.